For the Plant Phenome Journal webinar, we have Dr. Maria Balota. She's an associate professor at Virginia Tech at Tidewater, and she's going to be speaking on high throughput measurements of peanut canopy height using digital surfaces. Maria has been a very active member of the Tri-Societies, of which the Plant Phenome Journal is published with the Crop Science Society and American Society of Agronomy. And she's a board member and very active in helping move that society forward. So I've known Maria for a while. She's been also very active in the UAV community. And so that's some of the work that she's been doing on peanut, one of the innovators in this area. So Maria, I look forward to your talk. Thank you.
Thank you, Seth. I want to thank you for inviting me to be on the webinar today. And I'm really honored, and I would like to start my talk with acknowledging the co-authors of this work. And they are graduate student Sayantan Sarkar. He actually conducted the research and produced the draft of the manuscript. Doctors Bryce Kazanov, Joseph Oakes, David McCall, and Wade Thomason from the School of Plant and Environmental Sciences at Virginia Tech, and Lynn Abbott from Computer Engineering Department at Virginia Tech. What I would like to start with is pose you a question. With the changes that we are seeing in population, natural resources, and weather, doesn't feel that we need more resilient and nutritious crops to grow more so today than we did in the past. It looks to me like it does. And I want to show you that peanut is indeed a resilient crop. So the pictures that you're seeing here, sorghum at the left, peanut at the right, they were taken in the end of July this summer in southeastern Virginia, where we have 48 inches precipitation, annual precipitation, way more than you guys have in Texas. And I want to show you the resilience of peanut after, in comparison with sorghum, after 3 weeks of no rainfall on a very sandy soil, and with plentiful of moisture early on in vegetation, plants growing extremely well, and with relatively shallow root system. So not only that the peanut is resilient, but it is also nutritious. I know of a good number of research projects in Africa going on to combat malnutrition and food and hunger based on peanut products in children in particular. And they are saying that a handful of peanuts equal 1 chicken leg or 2 eggs or 2 glasses of milk or 3 corn ears or 16 servings of sweet potatoes. So it is nutritious, it is resilient, and therefore is growing on 21 million acres worldwide, and the production is 37 million tons, most of them coming from China, India, Nigeria, and United States. It originated from Peru or Bolivia, where it started to be grown thousands of years ago. And as my red arrows suggest, it traveled everywhere in the world. It moved to Africa, then to Asia, Europe, Australia, and North America. In the United States, it came with a slave trade in 1700s. And in 1800s was actually not grown commercially but on very small acreages. Like, for example, there is a document showing that it has been grown commercially in South Carolina, but nowhere else. Its value as a food was discovered with the Civil War, and soldiers relied on the nutritious food that peanut provided. Finally, in early 1900s, George Washington Carver started researching peanut in Alabama, and so real commercial production started. Arachis hypogaea is an allotetraploid with a genome form of AABB and A and B genomes coming from Arachis duranensis and ipaensis. It is a relatively short plant with leaves and flowers produced, of course, above ground. But after pollination, the the flowers produce special formations called gynophores that penetrate the ground, and in the ground, actually, the pods and seed are produced. Therefore, you may hear it under the name of groundnuts. The hybridization of it resulted in several subspecies and several market types, as I'm trying to show here, with a diversity of canopy architectures and seed sizes and peck sizes and colors. Virginia type is the largest of all, probably twice as big in pods and kernels or seed as the runner and Spanish and Valencias. Virginia and runners are mostly grown in the United States, but also some Spanish and Valencia as well. Yes, canopy architecture of peanut is quite diverse and is quite important for the morphology and physiology of the plant. There are four basic or main crop architectures: prostrate and spreading, and runners are within these groups. As you can see, the height of the main stem of those are probably anywhere at 10 to 20 centimeters. Then Virginia type, they usually grow as a bunch crop architecture with heights from 20 to 30 centimeters. And erect types, most of the Spanish and Valencia types follow this group with heights from 30 to 40 centimeters. Probably, I have not seen a peanut that is taller than 50 centimeters, period. Now, all these crop architectures, you can imagine then they can function different in terms of light capture. Photosynthesis and prostrate and spreading, for example, will have leaf area and spread in such a way that light interception is higher, so they can grow and cover the soil faster and more complete, and therefore reduce evaporation from the soil for the benefits of the plant. So therefore, water use of the plant could be affected by this architecture, but also being more in contact with soil than the bunch and erectophile types, they could be more prone to soil-borne diseases. So, and this is important because peanuts are good, are nutritious, are resilient, but they have numerous biotic and abiotic stresses that can happen to a crop if it is not well managed. There are several foliar diseases and soil-borne diseases that can be devastating for a crop. Basically, a late leaf spot can defoliate a crop in 2 weeks, and the same can happen with a crop be totally devastated by sclerotinia blight later on in the season, quite close to harvest, if measurements are not, cultural practices are not followed as they should. Numerous insects, caterpillars, spider mites in dry environments, but thrips are in particular interesting because they produce damage to the crop not just by themselves, but they are vector for tomatospotted wilt virus. Abiotic stresses, drought stress is a major one, and only 35 of the US peanut acreage is irrigated. In Virginia, Carolina region, only 15%. Crop growth can be stopped. Heat stress is increasingly becoming a problem. Pollen sterility, because of high temperature, has been shown to happen for many years now, but wilting under heat conditions start to become increasingly problematic in certain regions like Virginia, Alabama this summer, and we have determined that photosynthesis is impaired when leaves are wilted. Nutrient deficiencies as well. So in United States, peanut is produced on a little over 600,000 acres, hectare, I'm sorry, and in 11 states that are basically grouped in 3 main growing regions: Southwest, Southeast, and Virginia, Carolina. There are differences in the conditions rather than in management practices between the regions. Basically, peanut can be grown only on sandy soils. But for Texas, for example, New Mexico and Oklahoma, water is way less than the availability of water is way less than for the VC region. And also diseases and insects could be different from a region to another. What also differs these 3 regions from each other is the market type of the peanut that are grown within each region. For example, Southeast, they primarily grow runners. Southwest, they grow runner and Spanish type, and a little bit of Virginia type and Valencia in New Mexico. Virginia and Carolinas, they primarily grow Virginia type peanut, the big kernel peanut that really requires a lot of inputs and a lot of moisture to grow. For the VC region also, not just the growing conditions are different, but the breeding has a little twist, and it is different than from other peanut growing regions in the country. And basically, the breeding and release of peanut cultivars, Virginia type peanut cultivars, happens in 3 stages. Crosses and advanced breeding lines are produced at the North Carolina State University in its breeding program. And of course, they're tested for a number of years for yield, disease, and quality in the state. And then the lines in pipeline for a release, 20, 30, or 40, they are tested, they are moved to the peanut variety and quality evaluation project that I lead in at Virginia Tech. And we test those for a minimum of 3 years across all 3 states. And we provide information not just on yield and disease, but also a thorough analysis of the quality of peanut, including all shelling qualities, including seed oil profile, skin blanchability, pod breakability, and recently we included in testing evaluation under drought conditions under rainout shelters at Tidewater Research Center where I am located. After testing for 3 years, then, well, actually we have each year in March a PVQE advisory committee that includes members of all the peanut industry, the breeder, growers, researchers, shellers, seed processors, certified seed producers, all segments of the industry, and they are looking at the results produced in the PVQE testing, and we decide together what varieties to be released for the VC region. And the PVQE program, so which is a major component of my research program, is taking place in southeastern Virginia, where Virginia Thai peanut is grown, right, at the Tidewater Agriculture Research and Extension Center. And the picture shows 2020, a small part of the 2020 PVQE trials looking at breeding lines provided by NC State and the checks that we are using in testing, which includes current releases, cultivars like Sullivan, Walton, and Wynn. So what I was trying to show with this is that developing high-throughput phenotyping methods of screening seems to be a good fit as a important objective of my research project, and also a good exercise for a graduate student work. And here you go, Sayant and Sarkar starting the work on developing, estimating peanut height from digital surface and terrain models using RGB images taken by an UAV. And this is a paper that has been published in the Plant Phenome Journal. For this project, we used 104 accessions of the US mini-core peanut collection, including a diversity of market type and botanical types of peanut that were assembled and evaluated in Georgia, Texas, Oklahoma, and Alabama before, but they were not tested before under Virginia environment. That was the first time they were evaluated at the Tidewater ARAC in 2017. There are some things known about them from research on other regions, peanut growing regions, like growth habit, plant size, main stem height and characteristics, flower position, leaf colors, stem pigmentation, maturity, and also 3 major of peanut, leaf spot, tomato spotted wilt, and sclerotinia blight. But again, it is the first time that our contribution to how these 104 accessions are performing in the subhumid Virginian environment, it happened just now. We grew those, and the picture right here, you see the whole experiment that Sayantan worked on, including plots of all accessions of 3 by almost 1 meter size, arranged in a randomized complete block design with 3 replications. The methods we used, of course, we measured the main stem height. The image in the upper part of the screen shows the major growth habits, crop architectures that we have seen in this collection, spreading, bunch, and erect, and the height of the main stem, how it relates with the canopy architecture is quite clear. The height increasing from spreading, bunch to erectophile growth habit. We also noticed that there are differences in wilting and tomato spotted wilt virus on the plots, and we scored visually the wilting. Keep in mind that the wilting was not in response to drought. 2017 was actually an excellent year for peanut growth. It was in response to high temperature stress. And we evaluated those as well. Wilting and spotted wilt virus, they were not included in the publication I'm presenting today, but it will be in a different publication. Aerial imaging was with an Aztec octocopter and the Sony Alpha 6000 camera. And we flew in a waypoint navigation with proper overlapping and at a height of 20 meters above the crops. This table shows the dates and number of images when we flew the UAV, the dates and growth stages of the peanut when we took manual height measurements. They were pretty much at the same time. And what this information was used for, so 2 dates, June 19, June 27, were used to develop the regression model, and June 22nd to validate the model. And then the dates and growth stages of peanut when wilting occurred and when we took it, and also tomato spotted wilt virus counts were taken as well. For statistical analysis, we've used a JAMP and with t-test, correlation, and regression. Images were analyzed with 2 software. A Pix4D mapper we used to create the orthomosaic and also digital surface model and digital terrain model. And we imported the DSM and the DTM within the ArcMap of the ArcGIS. Now, this is the DSM created by Pix4D with color code elevations. And under D on this image, it is the DTM, digital terrain model, that was automatically created by Pix4D with a pixel size of 2.43 centimeters. And Sayantan decided that he's not very satisfied with that precision. And therefore, what he did under point B of this image, he actually created a shapefile, a fishnet polygons representing individual rows, and also he set ground points actually that allowed him to create a second digital terrain model under C in this image that was much finer than the automatically created one by the program, and with a pixel size of less than ½ centimeter. And by subtracting each of these C or D terrain models from the surface model, he came up with a height map, color-coded under E. And then he took each individual row, and you can see that the row, because canopy architecture is such that within each row there are several heights of the plant. You can see different colors on the image, but because we were actually interested in the main stem height, or the maximum height, which was what we measured in the field, actually he extracted only this information further on. So this is a summary of the results for the 104 accessions of the mini core collection, including market type, how the number of entries within each market type, and then the range and average for plant height, wilting score, and tomato spotted wilt virus. That was basically, this represent the numbers out of 35 plants per plot. So the ranges for plant height, for example, varied from 15 to 42. The wilting score wasn't quite severe as I would have expected in a drought situation, but it went from 1 to 3, pretty severe. And tomato spotted wilt virus out of 35 plants ranged from just 1 to 17, almost half. This is the regression that he used to estimate plant height from RGB images. And here on the x-axis, you can find the manually measured canopy height, and on the y-axis, the UAV-derived canopy height. And the upper images show the regression models when he used the ground point method, and the ones at the bottom when he used the automatically derived terrain model. All A's represent when he used all the data points, a little over 1,200 or so. And all the B images represent regression when he excluded outliers. And yes, indeed, we found outliers, and these are nothing else but plots, as you can see in the bigger image, where germination wasn't exceptionally good. And so we just had sparse plants within the row, and then the program, of course, included bare ground into making the average of the plant height of the row. So we excluded those and validated the model for accuracy of plant height estimation, A, with the ground point method, and B, with the automatically derived terrain model. And I think that shows a really nice accuracy of 91% and 78% of estimation of plant height from the RGB images. Now we also noted, and again, this has not been published in this article, but we will publish eventually, is that we found out that plant height is responsible 54% for wilting of the peanuts, and taller plants seem to wilt more under heat than the shorter plants. We also noticed that taller plants, regardless of the day of the flight and when tomato spotted wilt virus was caught, taller plants were having more tomato spotted wilt virus disease than shorter plants. So basically, in summary, we found out that peanut height can be successfully estimated from RGB images aerially collected at 20 meters above the crop, but uniform crop stand is important. We found out that both DSM models described efficiently and accurately peanut height phenotyping, but in the paper, Sayantan talks about when he thinks that one model is better to be used than versus the other. And I also think that we need to continue to validate the methods using this time real breeding populations that are have smaller height differences than actually the mini core. And also we need to validate the relationship between plant height and with biotic and abiotic plant responses in breeding populations. So I want to thank with this my phenotyping team. Of course, Sayantan and is the most important in this team, and also Bryce Kazanav, and also leading technicians, and Naveen Kumar, other graduate student that actually we are using his study in which he is looking at about 720 breeding lines from 3 plant populations to validate Sayantan's methods. And I will end up with thanking again the co-authors of the paper, as well as NIFA collaborators, because funding for this small project, I mean, this small project is a part of a bigger project with Texas A&M, Texas Tech, and Oklahoma State University, and the collaborators there, Rebecca Bennett, Mark Borrow, and Kelly Chamberlain, provided the seed of the mini core collection. And also I want to thank the sponsors, Virginia Crop Improvement Association, for funding Sayantan for this work, and the NIFA AFRI that also provided supplemental funds for research associates that did part of the work at this project. So I think that this is pretty much all I had to say, and from this, I'm happy to take any questions. I did not go to details about this work because Sayantan is on the line and I want him to provide those details.
Great. Well, thank you, Dr. Balota. Very good job. Nice cutting edge seminar in the area of phenomics for peanuts and measuring plant height. So we're going to open it up for questions for the next, we got about 20 minutes, 25 minutes. You can go ahead and write those into the chat and we can read them aloud, or you can unmute if you want to turn down your volume and ask your question after you introduce yourself. So while you're preparing questions, I'm just going to make a brief plug for the Plant Phenome Journal. That is where this study is published. And we are an open access society journal dedicated to advancing issues like the one presented in Dr. Balota's seminar today. And so I guess before or while we're waiting for questions to pop up in the chat, I have a number of questions, but I guess one of the most important ones is, so what's next for you? You know, plant height is, and you mentioned disease, but I think the sky's the limit in terms of other types of phenotyping you can do with this. What other things are you looking forward to using in your program, Dr. Balota?
Well, let me see whether I can share a couple other 2 slices with you to answer this. Yes, we do a lot of photogrammetry in our program. We are using color space indices such as hue, angle, saturation, lightness, and other parameters. Green area, which are calculations based on the hue angle. The same with the senescence index, reflectance in the visible near infrared and red to calculate normalized difference, vegetation index, normalized difference, red edge. We are also using thermal imagery to look at several other things, and some of them are just right here. We are trying to estimate biomass with Sayantan. He did nice work on leaf area index estimation. Actually, we submitted that for paper for publication, and it is in the review process. We also, right now, I'm writing with Sayantan, it is his work as well, estimation of plant wilting. I think that using machine learning, I think that that is quite an important one, because just phenotyping thousands of breeding lines, as you very well know, is time consuming. And if it is done remotely and slowly, it will be affected by the environmental conditions. So I feel confident that using the UAV will reduce that error. And we are doing work here in my program at the Tidewater Research Extension Center, but I have started timidly some work with a graduate student in a project, USAID projects in Africa. For example, looking at seedling vigor, for example, that they are routinely, the breeders are doing this routinely in their breeding program. Groundnut rosette virus and leaf spot are 2 major diseases in Western and Eastern Africa that I'm trying to work and find out tools, high throughput phenotyping tools, if you will, to can, you know, screening can be performed fast and as accurate as possible.
Okay, great. I see Dr. Anjin Chang unmuted. Did you have a question?
Oh yeah, hi. Oh, can you hear me?
Yep, we can.
Yes. The Dr. Maria Balota, thank you for the great presentation. So actually, I had a similar question with the Dr. Demore. So my question was, this like a research have been focused on comparing the peanut height between UAV based and the ground based measurement. But my question was, what kinds of the other phenotypes are strongly related to the some kinds of situation, like a yield or like the disease kind of things. So yeah, I think that the Dr. Balora already answered about my question. So yeah,
okay,
yeah, I have another question. So in this slide, the current slide, the platform looks like the DJI Matrix 200. So what kinds of thermal and multispectral sensor do you have in your center?
I just recently purchased, maybe a year or two ago, a DJI with Leica Sense camera. I have that as a new tool, so to speak. I am primarily looking at something simple, if you will, and easy to be implemented into a breeding program. So that is my purpose. That is my main objective for everything that I do. I think that if it is complicated, yes, I know hyperspectral, and I know that they are LiDAR, and there are other systems that can be used to phenotype. However, it is not something that I would like to play with it right now. I don't want to look into something that requires high technology and technological knowledge, if you will, but rather something simple. And yes, we started really simple. We wanted to see, can we estimate peanut height? I know this has been done for several years, maybe longer, in tall crops. But is it working in short crops? That is important. Leaf wilting, it is important. Sayantan had an additional test in which he's linking wilting with reduced photosynthesis and with reduced yield, because nonetheless, whatever we are trying to phenotype will have to have a response to yield. And I touched a little bit upon the fact that for peanut, the crop is in the ground. It is an indeterminate plant, and with good disease package that the new cultivars have right now, it is really difficult to say, decide when is ready and what kind of yield I'm having in this field. Biomass in Virginia is high. Sometimes it is just too much biomass, and the growers apply growth regulators to reduce biomass in order to see straight rows if they don't have GPS, and not everybody has GPS, to dig so that they will not cut the crop and leave it in the ground. So there are no other indications of yield other than what we are seeing on the ground, and that is wilting, that is plant height. That could be for a breeder. Plant height could be for a grower to pinpoint low spots in his fields where he needs to go and take a look and see what's going on. And this type of applications, I think that I'm interested in right now. Did I answer your question?
Oh, yes, thank you.
Thank you.
We have two questions in chat. One is from Navchat Kar. Dr. Balota, as you have shown us the work on plant height and its impact on TSWV, are you also considering relationships with other diseases such as leaf spot or sclerotinia blight using UAVs?
I was looking at the association between plant height and sclerotinia blight, CBR, and tomato spotted wilt together. And yes, it is a relationship. We didn't have much pressure either for sclerotinia blight for that year or for CBR in order to get more accurate relationships and explain the relationships if they are. We will probably need to do what the plant pathologists are doing infect the plants somehow, because otherwise we will rely on the disease pressure that exists naturally. And we didn't have for this particular application. It was almost absent for CBR, but it was some for Sclerotinia blight. And we have seen indeed that taller plants are less infected with diseased with a CBR, just like we expected, you know, based on theory and what I just told you about when we talked about canopy architecture. Now, tomato spotted wilt, why do I think that there is a relationship? I don't think that it is necessarily because of the disease. I think it is the thrips. That the thrips preferentially attack some plants and not the others. This has been known for decades. And for example, it is well known and established that they will prefer sparse plants rather than dense canopies. So I can imagine that when a main stem is just showing up nicely in the air and it doesn't have too much humidity in that microclimate, then the thrips will be on it, and with it, it will increase the possibility that tomato spotted wilt will be delivered to that plant. But it is just a hypothesis right now. We will need more money and more people to continue this work, but I'm really excited. I think we're definitely seeing something here.
Okay, we also have a question from Alper Adak. Is there any index color-based or NDVI-based you discovered that can distinguish disease symptoms and senescence at later growth stage in peanut?
Yes, actually, the greener area, and crop senescence index seems to be quite promising to describe defoliation from leaf spot. And not with drones, but with just regular cameras. I'm using those together with NDVI in my projects with graduate students in Africa to see what we can help with HTP HT phenotyping in those countries.
Okay, that's all the questions we have on chat. I don't know if anybody else would like to unmute and ask a question. I'll ask one more question while we're waiting here. So this is not the area you were trained in, right? So you've given a lot of credit to your employees, students, etc. How do you go about getting this knowledge and improving your program going forward? You know, what did you find most difficult about that?
Well, I guess I just need to extend collaboration with non-traditional plant physiology specialties. You know, really, I think that is very important that we have, like we had here Lynn Abbott with computer knowledge. I think that we need people that know machine learning languages, you know, that they can help us with making all these senses automatic as much as possible, so that we fly in the field and we can collect the data within a very short amount of time. In this way, actually, we could monitor the growth throughout the growing season of the plant, and we can estimate yield production much easier and more accurately than with the current growing models, I would think. So there are a lot of possibilities. I'm just a plant physiologist slash on occasion breeder or releasing cultivars, but their collaboration, I think, is very important. Did I answer your question? Is this what you wanted to hear?
I think so. I think so. Any other questions? Either unmute or throw them in chat. Okay, hearing none. Thank you, Dr. Balota, for a great seminar. Thank you all for joining us. And next month, we will have another speaker for the Plant Phenome Journal webinar series, who was recently published as well in the journal. And I look forward to seeing you then. Thank you all. Have a good rest of your week.
Thank you, Thad, and everybody.